So when you open, it's very simple, a cartridge inside.
So I'm going to be tested for the antibody for COVID-19 to see if I'm positive or negative. How does it work?
We have 5 different kits from different suppliers. They're all variations of the same theme. They're all kits that use a drop of blood to see if you have antibodies.
So which one are we going to use?
We're going to use this one.
What's this one called?
So it's just a rapid SARS-CoV-2. So first we've got to take a drop of blood, so I'm going to do that if you don't mind. So if you can just rest your hand, and what we're going to do is just clean the finger first, and then we're going to take a lancet, and 1, 2, 3, I'll try and be as gentle as I can, and there, we take a drop of blood.
It actually wasn't that painful.
Good. Just going to take it into a dropper, a small amount of blood, and I'll put that in the well. We'll do the same for the second well. For the antibody kit in a drop of blood, it is very much a case of a single drop of blood in a small cartridge, and we can measure whether your immune response has effectively tackled, neutralized and killed the virus. What we've seen over the last month is a lot of frustration with both from the government, from the scientists and from the clinical field. The frustration to get these out there and validated. It is very difficult to do. It's very rigorous and requires a lot of data, and there's just a difficulty in generating momentum. So we want to get some tests, validate them clinically and contribute to that.
Minerva Research Labs in New Bond Street is switching its medical expertise into virus testing.
And we can bring products into the market very quickly. We can analyze if they work. We have good procurement, we have good supply chain. What we understood from the testing, they are a bit tricky. Reading them is not easy. Sometimes the line that detects whether you are positive or negative is very faint. And to the eye of the professional who's used to see them many times, it would be very clear to say, yes, you are positive. But to the person at home, I think it wouldn't.
It's a new virus, and we just don't know how well protected, and particularly for how long. So if you have antibodies now today, for example, we don't know how long that immunity will last. We don't even know yet whether you're likely to get reinfected at another wave of the disease and exposure.
Okay, it's been 20 minutes and I'm going to find out the results of my test. So, doctor, have I had COVID-19?
Against the T line, there's no positive line, so you are negative. So the chances are you have not been exposed to SARS-CoV-2.
I guess I've been washing my hands properly.
Positive or negative, much more testing is needed. Rex Martel, ITV News.